Hey everyone, welcome to our staining techniques lab. If I can get it to click over, right? Sometimes it takes a little bit of new ones. There we go. So we stain bacteria because bacteria are invisible otherwise. If you look at a bacterial smear under a microscope, you're not going to be able to tell anything, a cell from anything else on that slide. They are invisible, and so we use stain to make a bigger contrast between the cell and the background. So bacteria are naturally negatively charged, and because of this, that negative charge allows us to use certain types of dyes called basic dyes that are positively charged. When you take something that is positively charged and put it with something that is negatively charged, they're going to be attracted to each other. And so you end up staining the cell itself. If we were to use a negative dye, so a dye that was negatively charged, then the dye would actually be repelled from the cell because the cell wall is negatively charged. And so acidic dyes are dyes that carry a negative charge. And so when we do a simple stain technique, we typically use a basic dye, which is positively charged. But if we're doing a negative stain, then we use an acidic dye, which is negatively charged. The two negative charges are repelled from each other, and so we end up having a stained background and we have clear cells. So you can find out two major functions or two major characteristics, that's a better word, about your bacterial cells using the simple stain and negative stain. You can tell the cell shape and the cell size, but that's all you can tell. We tend to use simple staining for most cells, but every once in a while we use a negative stain, especially if we're dealing with bacteria that don't withstand the heat fixing as effectively, or bacteria that don't take up dyes very effectively. And so my go-to bacterial species is Mycobacterium. They do not take in stain very effectively because they have a material in their cell wall called mycolic acid. And mycolic acid keeps certain stains from being able to be retained as effectively. They still will retain them, but not as easily as other cells. So and so this is the negative stain technique. In a negative stain, we take a drop of nigrosin, which is the acidic dye that we use. We add bacteria to the nigrosin aseptically. Then we use another slide to spread the bacteria from one side of the slide to the other side of the slide. We allow it to dry for about 10 to 15 minutes, and then we look at the bacteria. In a base or in a simple stain, I don't think I have that technique written down. No, I don't. In the simple stain technique, we take the bacteria and we put them on the slide. We heat fix them to the slide, and then we add a drop of basic dye. So the basic dyes that we typically use are are crystal violet, safranin, and methylene blue. Now we're going to get into some more interesting and more important stain techniques. These stain techniques differentiate bacteria based on certain characteristics. And so the first stain technique that we'll talk about is a gram stain. And so what the gram stain does is it separates bacteria based on the amount of peptidoglycan in the cell wall. Gram-positive bacteria have multiple layers, 20 or more layers of peptidoglycan, whereas gram-negative bacteria only have 1 or 2 layers of peptidoglycan in their cell wall. And so that alone is what the gram stain separates. So there are 4 basic steps to a gram stain. You take the bacteria and you're still going to aseptically transfer them to a slide and heat fix them. Once you've done that, then you're going to stain them just like you do a simple stain. You add crystal violet, which is a basic dye. Crystal violet will stain all of the cells. After 1 minute, you rinse with distilled water, and then you add iodine. This is the glue or the mordant that produces a crystal violet iodine complex. And what this complex does is it gets stuck in the peptidoglycan layers. Crystal violet alone can move into the layers or come out, but with the iodine attached, then the crystal violet iodine complex has a much harder time getting through the layers. And so when you have lots of layers of peptidoglycan, you're going to end up retaining that crystal violet color. After 1 minute, you rinse with distilled water again. Then you decolorize with acetone. I typically discolorize for about 30 seconds or until the slide runs clean. Typically 30 seconds is fine. And then you rinse with distilled water, and then we counterstain with safranin. Safranin is that light pink color. It's also a basic dye. And any cells that have been decolorized by the acetone are going to turn pink. And so those are all those gram-negative cells. So what we end up having is gram-positive purple cells and gram-negative pink cells. So here are the steps again. Application of the crystal violet, which will stain, oh my gosh, I'm sorry, which will stain all the cells purple. Application of the iodine, everything stays purple still. Then we have here the alcohol rinse, and in that acetone alcohol rinse, all the cells that are gram-negative decolorize while all the cells that are gram-positive stay purple. And then we have the safranin as our counter stain. So there are certain things I always like to mention. This is a great stain technique. It's going to be the most important technique you use throughout the entire semester. You'll probably do this technique multiple times compared to any other technique. But this technique can fail. And there are different reasons, like if you make a mistake, say you over decolorize, that will cause problems. If you mix up the crystal violet and the safranin, because crystal violet is so dark, it will cover everything and everything will look purple anyways. But a really big problem is usage of bacteria that are old. When we have old cells, they don't always retain stain as effectively. And so they can tend to give a false negative stain. So they look gram-negative, but they're actually gram-positive. So the best technique for making sure that your bacteria are positive or negative is to use newer cultures. Don't use cultures that are a week old. You know, if they're only a couple days old, that's the best way, especially when you're dealing with your unknowns. If you're growing your bacteria and you don't do a gram stain on day 1, you wait till day 5 or 6 to do your gram stain. You did other techniques first. If you're using that original broth, then you're probably going to get a potential false negative. That does not mean you have a gram-negative species. That means that you should quickly transfer it to a new media and then try it again. And so this is what actual gram staining looks like. So here you have these coccus-shaped chains of gram-positive cells and these rod-shaped cells that are gram-negative. Another technique is called the acid-fast technique. So the acid-fast stain technique is not as widely used, but it's very important clinically because it can identify bacteria in the genus Mycobacterium that cause very important diseases, specifically tuberculosis and leprosy. Mycobacterium tuberculosis and Mycobacterium leprae cause the different diseases, tuberculosis and leprosy. They have a special substance in their cell wall called mycolic acid. This is a waxy substance, and that waxy substance repels those basic dyes. Because of this, we use a special technique known as acid-fast staining to identify this species, or this genus. The acid-fast stain technique is a long stain technique. So it's one of those techniques that I told you some of your stain techniques are going to take you like 20 to 30 minutes. This is one of them. So you're going to, again, aseptically transfer your bacteria to a glass slide and then heat fix them. Once you do that, you're then going to take a piece of bibulous paper, just a small section of that bibulous paper that you blot your bacterial slides dry with. Take a small piece and that is clean and put it over the stain, your smear region. You then take the entire slide and put it on over a hot plate or a steam plate, which is filled with boiling water. Then you're going to cover the slide with an acidic dye called carbofusion. Carbofusion will sit on the bibulous paper and soak into the cells. You never let the paper go dry. So every about minute or so, you're going to add more carbofusion to your bibulous paper. You're going to steam the slide for between 10 and 15 minutes. I have steamed for 10 minutes on my paper, but you can go up to 15 minutes. Once you're done, you then remove the slide from the steam plate, and then you're going to take the bibulous paper and throw it into the garbage. You then rinse the slide using distilled water and then decolorize using acid alcohol. So this is different from acetone. Acid alcohol contains hydrochloric acid in it, a very small amount, but it does contain hydrochloric acid. You decolorize for between 30 and 50 seconds until the slide runs clean in general, and then rinse again with distilled water, and then you counterstain with methylene blue. Leave the methylene blue on for 1 minute. Sometimes I use it for 2 minutes because methylene blue is a little bit slower at staining. Rinse the slide with distilled water and then blot under bibulous paper and look at your bacteria. What happens is after that 10-minute period of steaming, you're going to have all cells are going to be this pinkish color. Then you decolorize with acid alcohol. The mycolic acid in the mycobacterium helps to retain that stain color, whereas the non-acid fast cells are going to turn clear. Then you counterstain with methylene blue. The non-acid fast will turn blue, acid fast will be pink. And this is what you'll see. So here we have all of the bacteria that are like coccus shaped here, labeled with an A, are your non-acid fast, and then you see all these pink rods, which are the mycobacterium. They are acid fast positive. The last stain technique that we're going to be working on, we may do a capsule stain depending on how much time we have, but the last for sure technique that we'll be working on is called the endospore stain. The endospore stain is a stain technique that allows us to identify cells that persist in the environment during non-favorable periods. So endospores are structures that can withstand extreme heat, dehydration, extreme cold, and they can persist for hundreds of years. An endospore stain technique actually helps us to stain those endospores so you can see them. When you do a typical stain on bacteria that contain endospores, the endospores don't take up the stain, and a lot of times you can actually see those structures inside the cells. They look like little circles inside of a colorful cell. But because there are also inclusion bodies and other materials that could be within the broth, we can't say that they are 100% endospores. So we do this endospore stain. The only two common, well, I should say the two most common endospore formers are Bacillus cereus, or Bacillus species, not cereus, and Clostridium species. Bacillus cereus is who we have in our lab. So again, we're going to do a technique similar to the acid fast stain where we heat fix, aseptically transfer the bacteria to a slide, heat fix the bacteria, and then we take the bibulous paper, put it onto the slide over the fixed region, add this time, or place the slide with the bibulous paper on the steam bath, and cover the bibulous paper with malachite green. We steam for 10 to 15 minutes, and again, you do not let that stain go dry. So you have to continuously drop more malachite green every about minute or so. Once you are done steaming for that between 10 and 15 minute period, you remove the slide from the steam, you remove the bibulous paper and toss it into the garbage can. You then rinse the slide with distilled water. We do not decolorize. The distilled water is the only thing we use. Distilled water will actually rinse the color from all of the cells. But endospores will retain that malachite green. We then counterstain with safranin, and the cells themselves will turn pink, whereas the endospores will stay green. And so this is a beautiful endospore stain. You can see the endospores, you can see the cells. So during this week, I'm gonna change this a little. We're going to start out on Tuesday doing the gram stain. You're going to do 2 gram stains, and each time you're going to use, in each of the gram stains, you're going to mix 1 gram positive and 1 gram negative species. Then on Tuesday or Thursday, we're going to do an acid fast stain. Each person will do their own acid fast stain. On Tuesday, you're going to do an endospore stain, but you're going to use, I only have, I believe, 2 bacillus plates, so you're going to be using those plates, and that means you're going to be sharing. With the acid fast stain, I only have 2 mycobacterium as well, so you'll be sharing. Then on Thursday, you're going to redo the endospore stain, and you're going to do the acid fast stain if you haven't done it. You're also going to be doing another gram stain to make sure that you have that technique down and you have more microscope work usage. Tomorrow as well, or not tomorrow, Tuesday as well, it is tomorrow, but I'm just saying it this way. Tuesday as well, we're going to redo the streak plate technique. So you'll get to have a chance to redo that and practice, okay? And then, of course, you'll have time to answer the questions, go over anything in your study guide, anything in the textbook, anything in the lecture that you're struggling with, okay? You guys have a great day, and I will see you on Tuesday. Bye.